So I'm excited to share some work we've done. I'm going to share some new ideas, I think, but in order to do that, I have to get you through some of the background thinking. And we're going to, in particular, talk about an idea we are playing with in our lab called ecological epistasis. And I think everyone's probably familiar with epistasis, which is when one mutation affects a different mutation's fitness effects. And we're going to talk about how maybe populations structure can affect individual fitness effects and how that might change the way a tumor evolves. And so feel free to share anything you like and interrupt me, please, if you want. And Ben, tell me how closely I should be worried about ending at 11:30 or at the half-hour mark.
I mean, if we can make it roughly half an hour, I think that if you're slightly longer, I think it's fine. But because we have one speaker after you, so
okay.
roughly half an hour would be good, yeah.
Okay, cool. So I think we all know, and in this crowd especially, I think we know that cancers and other complex infections and even populations of animal species can be thought of as complex and adaptive systems. They change, and we're going to talk mostly about cancer and infections today, but really they change over multiple timescales. And when it comes to understanding and maybe perturbing and predicting these different dynamics on these different timescales, it often can require quite different strategies. And so in our laboratory, we really, instead of focusing, I think, on what many folks think of in terms of cancer biology, which is sort of what mutations exist at the time I biopsy someone, we really try to think about the processes that lead to that. So in particular, these evolutionary branching or speciation events during which time tumors evolve and mutations occur. We also think about how the individual species within a complex tumor, so if you imagine this picture, all the different colors or different species of tumor cell, if you will, or specific genetic alterations that represent clones, how they interact with one another to shape and preserve that heterogeneity that we see. And further, we think about sort of what we can do now. So how the heterogeneity that you see encodes specific tumor-wide phenotypes in terms of drug response. And so it's these three timescales that I would say on the left is the longest, in the middle of the game theory is the medium, and then the blue is sort of the right now. It's how these three timescales play out. And I would say from the most clinical in the convergent area, what therapy works for this patient today, to the most basic science, which is how do we create and then preserve this heterogeneity through these different mathematical models. And we're going to focus on the two on the left today. Mathematical models of speciation and evolution and mutation, and mathematical models of game theory interactions or ecological interactions, and how they might work together. So to frame our thinking, we're going to first think about this first one. So where evolutionary branching points in fitness landscapes, we're going to talk about seascapes as well, and how models of these can help us understand things. And so the work today is going to be mostly done by a talented PhD student called Dan, who is a student of Sandy's, and I think maybe would have been at this meeting in a different world. And then Emily Dolson, who's now a professor at Michigan State, who is a postdoc in my lab, and then two other folks in my lab now, Ishan and Steph Owen, who you will see results from. But what we're going to think about is sort of how we can understand cell autonomous effects and how those things. So when I say that, what I mean is individual cell characteristics that cause them to grow in a certain way just by themselves. So this can include genomic changes, it can include transcriptomic changes, it can include many different things. And what we're going to do to think about this is we're going to invoke a common mathematical object to help us think about it. So imagine you have some space, in this case, the XY plane, that represents in some way your genomic alteration, a cell's genomic alteration or identity. And then on the Y-axis, on the Z-axis here, you then have the height, you have some notion of fitness. So this is very common. It's nice to think about maps like this. You go up hills and evolution takes you up these hills. Mathematically, this isn't quite correct because I think when we think about the genome, it doesn't work like a Cartesian plane, but instead it can work more like a hypercubic representation. And I'll show you some examples of this in a minute. And the first sort of set of ideas we're going to talk about is how one might use something like a fitness landscape in these spaces to understand the different cell fitness the differences in cell fitness within a heterogeneous tumor and how that might help us predict or even control evolutionary trajectories through some space. And so this is all represented in quite an old paper at this time, 2015, but I think it helps to understand the thinking step by step. Again, this is mostly work done by Dan, who's cleverer than I am. And we got an error number of 3 because of this paper. So how do we take that idea of that topographic map, like we like on the right, which is intuitive to we who live in 3 space, how do we take that and put it into something that better represents the genome? So what we can do is we can take these ideas of these sort of higher dimensional Punnett square-like objects, which are just alterations in single loci. You're either a 0 or you're a 1, you're wild type or you're mutant at each loci. So it's the simplest Punnett square is that little square you see on the far left. And that says, you have long ears and blue eyes, or short hair and brown eyes, and then you can have any of the 4 possible combinations. You can extend that to arbitrary numbers of dimensions, and you end up with 2 to the n, a bit string of length 2 to the n. And then if you write down some sensible fitness mapping f, you can map whatever genomic identity you have of zeros and ones to some number, and that number could be your fitness. And so instead of this Cartesian 3-space surface you'll now have a sort of higher dimensional surface, like on the lower right here, you'll now have a series of corners of a hypercube that each have a fitness attached to them. So you can see each of those corners has a fitness, 0.1, 0.24, whatever it is. And those can be measured directly, right? So each of those corners of that cube now represent an organism, say, with a very specific genomic identity. So you can see in the middle picture, there's just a bar graph of fitnesses. So for each of the 8 different corners, 2 to the third, so 0, 0, 0 on the far left, that's the wild type, and it has fitness 0.73. You can see that sort of encodes that far left corner in the hypercube. And so what you can do is you can write down what looks like now a directed graph. You can say, okay, evolution goes from low fitness to high fitness. So for example, if I started at 110, which is that upper right corner at fitness 0.1, you could imagine that you could go up in one of 3 different directions. Any direction is up. Those are all uphill walks. And so you can write down a directed graph that kind of lets you understand which way evolution might take or an individual or the population as it moves around on this surface. And so thinking about in this way, we can sort of model these evolutionary trajectories as a random walk on this directed graph that we could measure in theory. You could take organisms with each of these different genetic aberrations, you measure their fitness and go forth. And what you can do then is a nice thing. You can write down discrete time population dynamics simply with matrix multiplication. So now you can really have the entirety of the evolutionary process encoded in a probability or included in a matrix of probabilities of transitions between those points. And through, you can then understand how these populations might go from on the far left, you see evenly distributed at all genotypes. And as you move through time, you're starting to see the individuals move up the peaks. And so those 2 peaks there are the same as the 2 peaks you see represented here that are absorbing. So on the far left, 0.73, and on the far right at 0.49. If you follow the arrows, you'll see that those are the 2 peaks and they end up absorbing the populations. And that's what we end up seeing with sort of simple matrix multiplication and a very simple mapping of fitness to very simple genotype to phenotype mapping. So the big idea from this paper was if you had more than one of these landscapes and they represented drugs, you could imagine applying them in different orders. So maybe in this case, this picture is showing you have a population distributed across all possible genotypes in the case that there's only 16 possible, which I think everyone knows is too few. But if you apply a drug called ampicillin, for example, you'll end up with a distribution over 3 peaks because it has 3 peaks and they're all accessible. If instead you gave a drug called SAM first and then ampicillin, so you went to the right and then up and right, you actually end up with only a single possible evolutionary outcome. And further, if you gave a series of 3 drugs in a row, you end up with a single evolutionary outcome, but just one. And so you can see that sort of through this collection of orderings, we can, at least with a Markov model, predict and steer populations to specific points in a genotype space. We could create a distribution that we like. That's all in theory. And if we try to do this in the laboratory, some problems pop up. So what we did in the lab is we just did the experiment. We took the exact same E. coli We did actually 60. I only put 12 here, but we did 60 versions of the exact same experiment. We just put them in the same drug, and one might expect, as one might expect, they all became more fit over time. So the evolution of resistance occurred. And the problem is, or the good thing is, they all became more fit every time we did the experiment. So X1, 2, 3, there, as you see, are all different experiments, and those are the fitnesses over time going up. But they're all quite different. There's some that are similar, and they all go up, which is a good thing. But maybe they're going up lots and lots and lots of different hills. So I guess the question is, I think the picture I showed you at the beginning was 2 hills. That's probably not enough. But how many is enough? I don't know for sure. But we do know that it's not as simple as the math model I showed. If we look at this evolution in vitro, we see many different trajectories, again, all monotone up, but all with very different consequences. So if you take a slice in time at the end and take all those bugs, so these are all now bugs that started exactly the same at P0, and now at passage 10, so 10 days later only, they're all now very different collections of bacteria. And in particular, they have very, very, very different drug sensitivity profiles. So if you just take 4 different drugs and ask how do they react at the end, remember, these all came from exactly the same populations. These populations all respond very differently. So you see just a little heat map of the change in response to these 4 very common drugs, PIP, ampicillin, ticarcillin, and so forth. And so I think what we're seeing is that there's evidence, at least in vitro here, for these divergences, for this idea that evolution is occurring, but really it's all going uphill, but it's not happening in the same way every time. And not only is it diverging, but it has vast evolutionary consequences. And so it's important to understand where it's going and why. And then further, like I said, I think two peaks is not nearly enough. I don't know how many is enough to do this, but there's also another problem here, is that these things might not be stable in time. So the little picture I drew for you of this little map with 2 peaks, it really doesn't necessarily have to be that simple, and it likely isn't. It's likely that these things change, especially with drug dosing. And this change in topography, so what you're seeing on the top here is 2 different drug response curves. On the upper left, you're seeing sort of a naive drug response curve that expects that the landscape you measure, which is shown in the middle, is the same rank order-wise, no matter what the concentration of drug. And on the right is a realistic, but in this case, made up drug response curve that shows significant evolutionary trade-offs through time. And you see here, if you look at the red trace, you see this cost of resistance, which I'm sure has been talked about at this conference. So on the left, with very little dose, this red mutant is the weakest, the lowest fitness. And on the right, at the highest dose, it has the highest fitness. And so you end up with these evolutionary trade-offs through time. And if you look at the bottom row, what you're seeing is just an example evolutionary trajectory in a scenario where the gray trace is sort of a slow, stepwise increase of drug. Now, why do we care about that? We care about it because the dose in a patient that you have isn't a stable thing. So this is just a silly mathematical model of pharmacokinetics that shows that if you take a pill every day, you still end up only with a dynamic equilibrium, never even a steady state. And if your patient forgets drugs every once in a while, the concentration that they experience in their plasma goes all over the place, not to mention spatial differences, not to mention drug delivery differences. So now we have this scenario where patients are going to experience wide swaths of drug concentrations through time and space within their tumors or infections. And if what that means is that the mutant that is most fit changes on the course of days, hours, and even longer and shorter timescales. And so this is a sort of scary prospect, because if this is truly the case, the drug we're giving, which kills something that we think might not be doing that all the time, might actually be promoting resistance in the morning, and in the evening not. And again, that's sort of those were cartoons, but this really does play out in the real data. So on the left is some data from a friend of mine called Brandon Ogbunu, who's at Yale, who showed this in a beautiful paper in 2016 in a transgenic yeast species, where these trade-offs really play out through time. And this is now in a 4 cube, so 2 to the 4, which is a beautiful object for the Marvel fans called a tesseract. So you have 4 different loci, and you can see these trade-offs abound. And on the right, this is data from our lab where we did the same experiment, but in a different kingdom of life in E. coli. looking at cefotaxime, which is just a common antibiotic. And again, you see evolutionary trade-offs all over the place. And so depending on what concentration you measure the landscape in, you can see those landscapes on top, the rank order changes wildly. And it changes on even over small periods of drug concentration ranges, and ones that are clinically meaningful. So now we have this problem. So now we have a problem that I think I've shown you, I hope I've shown you, that we can think about these landscapes and we can think about biasing evolution using them. But the problem is they change continually in something like drug dose. So our Markov model will no longer work to help us think about this. And so what we did was we came upon a better model, I think, one that allows us to think about how things change on constantly or continuously varying surfaces. And so in this case, we're going to invoke a population genetic model at its heart, but really one that's based on PDEs and thinking about something like a Fokker-Planck formalism. So I'm going to show you a couple of cartoons of how this model works. We now have 3 energy surfaces, the blue, orange, and green, with different selection strengths. S is minus something, S is 0, S is 0.1. And on the bottom right, you can see what the population distributions would look like after evolution happened. So for the physicists in the audience, put a ball and drop it on top of one of those surfaces on the left, and then let it wiggle a little with some Brownian motion, and its position will be represented by the distributions on the right, depending on the fitness that you have. So blue in the middle, orange on the left, green on the right. And this is all really done with a simple PDE. Looks like a reaction and advection, and you end up with something that is now directly solvable and understandable in a meaningful way with a minimum of parameters. So just here's some cartoons of how this looks. So if we have, what you're seeing is the green line that's moving is the time. And as you see on the bottom, what you have is the instantaneous distribution, which is the blue, and then, sorry, which is the green, and then the blue is what your predicted final distribution would be. And on the top one, what you're seeing is the change in selection strength from minus 0.001 to plus 0.001. So let's imagine that a patient is taking a drug that moves the selection from bias to the left to bias to the right. And what you'd predict is you'd say, oh, he took this patient, he or she, or they took the pill. So now we have this new selection. But what's happening is that population is sort of very slowly approaching that predicted equilibrium. Now, sometimes this is thousands of generations that it takes to reach there. So thousands of generations for a tumor cell is years. And so if you're really trying to affect the population, this is a difficult proposition. So what we propose is we propose a different methodology, which is something called counterdiabatic driving. I'll give you some intuition for in a moment, where instead of changing the drug dose, so that blue curve on the right now is the same exact blue curve you saw on the last slide. Again, you're taking the pill, you're minus 0.001. At the end, the new selection is 0.001. And the red trace there is now a corrected dosing schedule, where we have the same starting dose, minus 0.001, and the same finishing dose, positive 0.001. But in the interim, we give a much higher dose in time. So we give this external control field, as the physicists would say, in which we sort of bias more drastically this population. And what you see on the bottom is that this actually ends up taking the red distribution, follows in perfectly the predicted distribution in time with no lag. Whereas the blue one, which is the same as the slide before, moves very, very slowly. And this really allows us, this formalism really allows us to quickly and accurately control these evolutionary trajectories. And the intuition for this is like if you're a waiter, and you're taking water to 2 or 3 different people in a restaurant. Of course, you want to get there quickly to get a good tip. But you also, the risk of moving quickly is that you spill the water. And so everyone who's delivered water on a tray before knows that if you want to move fast, you sort of tilt the tray forward a bit and the water won't spill. This is the same idea. We're really putting in an external control field to be able to get somewhere quickly. That's fine in one dimension. What if we used some real data? And so this is again, work by Mike Hubchesky and Emily Dolson and a woman called Shamreen Iram, who's at Caltech now, where we looked and we took those data from the Ogbunu seascape I showed you and asked if we could use those specific data to do the same, if we could actually use a real genetic landscape rather than that fake 1D I showed you to drive these systems at arbitrary speed. And so here you're seeing a trace. So the dotted lines are where we expect things to go before and after drug. And the solid traces are that long lag. So you actually see about 1,000 generation lag between 500 and 1,500 before we get to where we think we're going. And if we're able to do this through time, we can apply our driving protocol. Uh-oh. The clock has stopped. Am I frozen? Can people hear me?
We can hear you.
Oh, good. Sorry, something happened on my end, but it seems fine. If you then apply this driving protocol, you can see that you can snap the distributions where you want them to go. So that's all very nice. In a theoretical system, it seems like we can quickly and accurately drive populations through these trajectories if we have knowledge of these seascapes. The problem here, I think this is all, I'm very excited about all this work, but the problem is, and I'm about to show a slide that's actually Sandy's. The problem is that cancer is not just a collection of isolated cells that are doing their thing. So this idea that I can just measure each individual type of cell and use that growth rate in some model to go forward is a bit naive, right? We really do have instead a beautiful ecosystem of cells of different types and even normal cells that affect one another in meaningful ways. And one way to call that is a non-cell autonomous effect. So cancers can have their individual growth rates driven by microenvironment, by other parts, by other cancer cells, and fitnesses of individual cells, which I was showing you beautifully as these little traces through drug dose, can actually be frequency and context dependent. So that sort of begs the question, does the structure of the population affect individual cell fitnesses? And does that really screw up all that control stuff I showed you a minute ago? We're going to just briefly talk about something I'm sure people have already introduced, this idea of game theory models. Here's a beautiful paper by Dr. Gatenby looking at how cancer evolution might be modeled in this way using a formalism called Lotka-Volterra, or you can also use replicator dynamics that ends up being similar. People have been using this in ecology for long periods of time. And in particular, this is really the first thing I did as a student with Sandy and David. And really what you do is you write down a dynamical model. You have a payoff matrix that tells you about how things interact. And then over the period of time, you can now figure out what the population traces look like. So these are the same sorts of graphs I showed you earlier. And what we figured out in a range of papers was that intervening early changes the overall evolutionary outcome. So if you give drug at time 10 as opposed to time 30, you have very, very different outcomes. So when you treat matters, even if the same underlying mathematical model exists. And also how long you treat matters. If you give a drug for a little bit or a lot of it, you might have green winning on the lower left or blue winning on the lower right. And all this really depends heavily on, of course, the underlying mathematical construct. But again, these are the same cells, if you will, doing their things. So in the form of my first models, this shouldn't really matter. The same thing occurs. You can look at when you treat, and there's a different paper by Artem, sort of same idea. If you know the underlying mathematical structure, you can answer all kinds of questions. But really, does it matter? And I think that the group that you're within now, in the room you're in, has really shown beautifully that it does. In a series of clinical papers that have been written by the group at Moffitt, I'm sure have been presented at this conference already, we really see evidence that these ideas of thinking about evolutionary dynamics and treatment can help you shape and perturb these tumors in a different way. And this is without necessarily knowing perfectly the individual fitnesses of the genome that I showed you before. But it does require knowing something about the structure of the matrix. And so to answer this, me and some friends designed an assay to directly measure these matrices from competition experiments using a specific kind of non-small cell lung cancer. The important thing to remember here is it's a quite contrived experimental system. And really the takeaway from this is that not only do we see dynamics changing when it comes to context, but we also see population structure directly affecting dynamics. And so in this plot here, which we call a game space, what we're seeing is how the different dots there. So look for example, at the two purple dots. What those are showing is what happens if you give a drug called alectinib in the presence of fibroblasts, so kind of like a very real-ish scenario in vitro. And you see the dynamics changing from stable coexistence in the upper right, where resistant and sensitive cells can live happily together. That would be in a patient before treatment. To if you move down into the right, the alectinib plus CAF payoff matrix, you really end up with a scenario where only the resistant wins out. And this is sort of what we see in an MTD kind of scenario. So thinking through this, we want to think about this as a different knob to turn. And going quickly through, we've also now seen the games change widely with the dose of the drug itself. So in the original paper, we just showed that adding drug helps, but it turns out that these dynamics change drastically even as you increase drug dose. And further, if you take the same exact cells and measure what happens in collateral scenarios. So now you take cells that have evolved resistance to a first drug like gefitinib and put them and compete them in a second drug that they've never seen. In this case, lapatinib is cisplatin and osimertinib. What you see is, again, these dynamics move all over these charts. The takeaway here is that these competition dynamics move through 3 different quadrants in some cases, where we have very different evolutionary dynamics. Remember, these exact same cells. So nothing is happening. There's no mutation. Well, there could be mutation in theory, but this is a very short assay. This is a few days. This isn't hundreds of generations. And so I think the takeaway is that in the context of changing drug dose and evolutionary context, both your cell autonomous and non-cell autonomous fitnesses change. And so this is a problem because I'm really hoping to be able to control some of these things. The good news is these things are measurable, but right now with the formalisms, the mathematical formalisms separate, we're in a little bit of trouble. So what would happen, and there's only 3 more slides, I want to have some time for questions. What would happen if we tried to decouple these, what if we tried to combine the 2 formalisms by smartly decoupling these things? And so this is work from Steph Owen, who's an excellent postdoc in my lab. And what they've done is really think about, so what does that cell autonomous, the no frequency dependent situation look like? Well, it really looks like a payoff matrix like you see in the lower left, where no matter what type you are, you have the same fitness. So if you're the top row type, you have fitnesses A and B, and then that matrix down there, it's the same 1 + S1. And if you're the other type, you can normalize the fitness to 1. And so that's sort of the very, very quite boring version that I've shown through all the fitness landscape stuff. But now if you put in, instead of having it written down like a payoff matrix, like I've shown for the game theory models, you can instead put in these interaction terms directly and maintain the monoculture fitness within the matrix. You can now actually start thinking about this in a different way. So the question that I'm posing is what happens when you combine these formalisms? And the answer is that sort of all hell breaks loose. And what I mean by that is that for any version, so let's say you measure monoculture fitness, and you think I have 2 types of cells, type parental and type resistant, and you figure to yourself that one is much more fit than the other, like in my initial simple one-dimensional example. And now I want to see what happens when they compete with one another. But it turns out I can get almost any evolutionary dynamic I want based on the interaction terms. Now, mathematically, that's not super surprising because of the way that linear algebra works. But I think biologically, it's a bit scary. Because if we're making monoculture measurements, what that means is we really have no idea what's going to be happening in competition. The good news is that this formalism is now also amenable to the control protocols that we developed previously. And further, once you start measuring these things, you see, and we have this idea of mimicry, masking, and maintenance. Maintenance sort of means that what you measured in monoculture is actually what happens. Mimicry is the opposite of what you measured happens. So you measured type A being stronger and B being weaker, and you end up getting the opposite. And masking is that you have no idea what's happening at all. But there are underlying patterns in the dynamics that can give us a hint as to what's going on. So I'm not completely at a loss. And further, we can now measure all of these things. And so this guy in my lab called Dagim is now constructing the first hypercube built out of cancer cells. And so we've used transduction to create the first square, which you see there, which actually includes the 0000 case is a wild type PC9, which is a kind of non-small cell lung cancer cell. And then we're putting in combinations of mutations in EGFR, BRAF, and KRAS to create a full square. What we're seeing now is that we do have the expected fitness differences in monoculture, and we're waiting to see what the games look like. And so in particular, in the upper right, you can see our sort of experimental plan, which we've now measured the square, the seascape in these cells, but really understanding how they interact with one another is going to be key to the overall dynamics. And I think that the key takeaway is that we really need to be thinking about these interactions if we want to have any version of control. But measuring these things thoughtfully, I think, is going to give us a way forward. And that's all I wanted to say. Thanks to the group. And you can see the smiling faces of a lot of the folks that contributed to the work I showed in the picture at our last retreat. And also thanks to folks that have funded our work. And that's what I wanted to say today.
Thank you, Jake. Do we have questions for Jake?
Jake, howdy. It's Joel sitting here in the back. I'm not sure you can see me, but that's probably a good thing.
Hi, Joel.
Hey, Jake, great to see you. First, observation number one, I love seeing the chess set there, the game theory right there on your desk. That, yeah, see if I can do pawn to king 4.
An IMO, sure.
Yep. And then the second question, you emphasize the importance of having frequency-dependent selection. But earlier when you were showing us the fitness landscapes, you were considering them to be fairly rigid. Would you like to kind of comment then on that sort of rigid landscape versus a very frequency-dependent view of the world?
Yeah, absolutely. I mean, I think that's the whole point that's the whole point we're working on now. I think that the fitness landscape view of the world is mathematically very fun to play with, and it's tractable. You can write down solutions to the evolutionary problem. You can measure these things by themselves, and it's even amenable to control protocols, which is very exciting. But the more and more we measure these things in competition, the more we realize that frequency dependence is the rule, not an exception. And so I think really that a lot of the fitness landscape work that I and others have done should be taken with a grain of salt, because I think that any predictions we make about temporal dynamics using those models is likely incomplete without taking into consideration frequency dependence. And so in particular, I think, as I was showing at the very end, I think that what the models we're choosing to work with going forward are really the combination of the two. And I think the clever mathematicians in the room who are there, likely, you could say, well, how is the combined model you show at the end any different than a regular game theory model? And that's an astute observation, in that they are in some ways very much the same, or can be decomposed into the same things. But I think that by decoupling the two and continuing to consider the monotype measurements that you can make, it really allows us to measure the individual components of the model rather than only being able to consider the phenomenon on a sort of mesoscale. And so the excitement I have is that we're sort of finally now able to dig into the model to measure the things we think we need to measure to make sensible perturbations.
Hi, Katarzyna, behind Joel, even more back. So in your ALK paper, you have these fully resistant cells, and I think it took like some months, let's say, to make them fully resistant. And they have quite special properties, right? There is not cost of resistance, there is more like a gain of resistance in a sense that they grow much faster than the sensitive cells. Isn't that possible that during creating these fully resistant cells, you selected for the strongest clone? So meaning there were many clones which would be resistant, and the one which you do your experiment with is by chance the strongest one, the fastest one, right? Because that one, you gave it, you said 4 to 6 months. I thought it was even 8 months, but okay, that's it.
Yeah, no, you're bang on correct as well. And I'd like to emphasize that the second set of data I showed was from a different set of mutants in EGFR lung cancer. And that paper is actually now, you can find it on the bioRxiv, but it's in press and Science Advances. It's Farokian et al., and I'll send a link. And that paper is in a totally different context where we evolved gefitinib resistance. And I think the most important thing is that I think if I had done those initial evolution experiments again, or 10 times, just like we did for the evolution experiments in the bugs, if I were to do the initial evolution experiment to derive the resistant cells and then measure the gain, I don't think I'd get the same gain every time. And so you're absolutely right. In the ALK paper, the first one, we have a very strange scenario where there doesn't seem to be a significant cost to that resistance in monoculture. But in the EGFR paper that's to follow, there actually is a significant cost to resistance in monoculture. And what we're working on now is sort of combining those two ideas to say, what happens if you did repeated evolution? So let's say I took the ALK positive cells and evolved 10 replicates of resistant clones in totally separate experiments, and then measured each of those 10 gains. What do they look like? And in the EGFR paper that's just coming out, we show that it looks like to be a KRAS and MET-based evolutionary strategy that beats the cafitinib. But again, that's just one instantiation of evolution. And so I think instead of focusing on the result itself, which is to say that, you know, this type I evolved has this very specific example, and therefore every time in ALK you're going to have this game, I don't think we're trying to say that. I think we're trying to show a method by which to measure any game. And I certainly think that if you do the experiments again, you'll get different answers. I hope that maybe those answers will fall into a statistically understandable ensemble, but I can't prove that yet.
Cool. Hi, Jake. Great talk. I think you kind of touched on my first question there, which was, you hinted at this non-repeatability of evolution and whether that would actually render a lot of those payoff matrices kind of so stochastic that they wouldn't be tractable. So my other question is probably maybe a bit more tedious, but it's like experimental. So you kind of, you do these competition experiments where you're mixing ratios of resistant cells in culture, and presumably those are all in 2D. So I just wondered if you could comment on how, you think that the competition's actually operating on a kind of 2D layer level. Do you think something's being used up in the media? And then, therefore, what are the implications for how that translate into, an actual 3D tumor?
Yeah, really good question, and I can't see who that is. Is that Alistair? Who's that?
No, this is Freddie. I'm a postdoc in Trevor's group now.
Oh, hi, Freddie.
Yeah, nice to meet you.
Nice to meet you. Nice to meet you too. So I'll rewind the clock a little. So in the ALK paper, the first one, we think that it's a diffusible hepatocyte growth factor based mechanism. We're not sure, but we think. There's some good evidence that Andrei Marusek has in his lab that that's the case. But again, I think that's just one evolutionary solution. We're excited. One thing we're excited to do is get back to my roots a bit in agent-based models and start asking the question, what range of diffusible phenomenon could supply the phenomena we see? And we don't, or do you need an infinite diffusion length, or is it a close one? And like you said, these are 2D experiments, and the data that we're analyzing are cancer cells sitting down on a plate. And so the spatial data do exist. We just haven't used it yet. We've made some attempts, but it turns out it's hard. And there's a clever woman called Sarah Hammes, who's a Leonard Gren fellow in Finland now, who's working on some of these ideas with us. There's a beautiful preprint out now about spatial cumulant models, which I recommend people look at. But I don't know yet how to parse that spatial information, which is what I would need to do, Freddie, to get to your answer about 3D. And I have no idea. I've also put a grant in 6 times in a row now to study that specifically, and the NIH has not yet deigned to fund it. I will say it's also at study section today again. So if you have any friends at MABS, call them and say, hey, there's a really important question Jake should answer. And he can only do it with your help. So vote for ones.
Hi, Jake. Katarzyna again. Okay. No, I had just an additional question to my previous question. When you were creating these fully resistant cells, did you somehow sequence them? Because that would be very interesting information, right? To see these different clones for any of these experiments. Yeah.
Great question. The answer is no. So we bulk sequenced the ones in the second experiment. And I think it was useful, and I will say that in the shower this morning, I thought to myself that it would be really interesting to do single-cell sequencing on different evolutionary replicates in these competition experiments. In particular, I don't know, so one of the things we need to do to make this assay work is measure things at different concentrations, so like 2080 versus 8020. And now, I think we've shown convincingly that they have different fitnesses in those different contexts. So I'd like to know even if the sequencing and transcriptomics are different for the types based on the frequency dependence. So not only the question would be, is there frequency-dependent fitness effects, but are there frequency-dependent transcriptomic effects that drive that fitness effect? Or is it simply is everyone doing the same thing they would normally do, but because of diffusible factors like Freddie was saying, it works out? And I don't know the answer to that. I think that that's answerable because I think you can multiplex out the red and green because of the fluorescent proteins and then do a single cell analysis, but we haven't done it yet, but I'd like to.
Great, thank you. Maybe in the interest of time, we should move to our last talk. Does that sound good, Ben?
Yes, absolutely. Thanks, Jake, again.
Thank you, Jake.
No worries,
Thank you.
Sorry to miss you. Have a beer for me.